But finding where the cell is only half the problem. To understand how we track a cell, we also need to understand what the model thinks a cell looks like, right? So what exactly represents a cell? Before that, I want to clarify something in our last video. A UNet is supposed to find candidate cell locations, right? But our current movie is biased. It has a lot of cells in one region. And if a lot of our sample is biased in one axis or in one region, our model might give biased answers. And because we don't want that happening during training and prediction, what we do is we flip the images in X and Y and we orient the image in multiple directions so that cells, on an average, spread out. The model does not learn to be biased with regards to giving its predictions. And then we average out the logits and get the best prediction. That's a side note. But what exactly does this model represent as a cell, right? What does it reason about? Remember those unit feature maps? That is what a model thinks a cell is. Those are dense representations and they are for every point in the video. But we don't need to give the next model every pixel, right? We have identified where the cells are. So we just want to know what's around that cell. And the unit feature is 32 dimensions of what the model thinks are features around that cell coordinate. So for that, we detect each cell, we sample the unit feature map at that precise location. It's like putting a pin on a cell and asking what is important around this point, all right? So that gives us a feature vector, and that applies to each and every cell. For each and every cell, we get different feature vectors. But just knowing how a cell looks isn't enough, right? We also need to know where the cell is. So what happens is we also encode the four-dimensional data, time and x, y, and z positions, as another vector, which is just to say we note that down. But instead of just having 4 positions, we have 32 positions. Now these are sine cos transformations, which very simply put tell the model in different resolutions where the cell may be. Now this is complicated stuff, but it's 32 dimensions that exactly encode where the position of a cell is in different in intensities or in different clarities. And that is what we input to detect different cells, right? So now we know what a cell is, how a cell is represented. It's 64 dimensional data that gets linearly transformed into 128 dimensions. But how exactly does the model figure out which cell belongs to which cell in the next frame?